So yeah, today I'm going to talk to you about a couple of examples of where we've used spectroscopy for food safety applications. No good talk about spectroscopy is complete without the electromagnetic spectrum, because spectroscopy is all about using light to study samples. So you're looking at how light interacts with matter. And the visible and the infrared regions are my favourite regions, because that's where the types of spectroscopies that I use, that's the light that we need. And so I am involved in a couple of projects, and we use the two main groups of spectroscopy, vibrational and electronic. And chemists and physicists are not very imaginative when it comes to naming things. So vibrational spectroscopy, as you can probably imagine, involves vibrations of something. And in this case, it's molecular. So we're looking at how the nuclei of molecules move relative to each other when we inject some energy into the system. And the energy we inject into the system is light. And so what we're looking at is how is light scattered or absorbed when it hits our sample, and from that we can get a lot of chemical information. Vibrational spectroscopy comes primarily in two forms, Raman or infrared. So a lot of people involved in the food industry probably have heard of near-infrared spectroscopy, because it's used a lot for food quality and control. Raman spectroscopy, not so much, but I'll give an example towards the end of my talk where this can be quite useful for adulteration studies. And why they're attractive to the food industry is because they're rapid, non-destructive, and there's a whole lot of different types of experimental configurations you can use to get the information from your samples, so that you don't have to do a lot of sample preparation. This is why near-infrared spectroscopy has been used a lot in the food industry, because you can basically have food samples going through near-infrared light above and collect your signal in seconds. And you can look at multiple things at once. You don't have to have a separate test for each chemical often. In terms of electronic spectroscopy, what I'm interested in is fluorescence spectroscopy. So for those of you who have not heard of fluorescence, it's basically you use light of one colour, you irradiate your sample, and you'll get light of a different colour, or different wavelengths. And so that's what you see in an example here in just a piece of calcite. And depending on the molecules involved and what you're looking at, you need to use different wavelengths or colours of light to irradiate your sample, and you're observing different colours of light. So in this example, these are just a whole series of proteins. They're all under the same UV light, but they glow very different colours because of the different chemistries involved. And just to bring it back to something that's related to food safety, this is just bacteria that have been stained, and they're under blue light, and they glow green because of what they've been stained with. It's called electronic spectroscopy because it involves electrons. So I'm a chemist who hangs out in a physics department, and I'm now attending a food safety conference, so it's a little bit odd. And it gets even odder because it started out with meat. So as most people probably know, in New Zealand we have very strict sampling regimes to test meat in the abattoir for total viable cell count. Meat gets swabbed after it's dressed. Swab gets put into, or 6 swabs in the New Zealand sampling regimes, gets put into peptone water, either gets couriered to a lab somewhere, or it goes to the lab on site, and they do a standard plate count to get total viable count. And this gives information about whether there needs to be a factory-wide alert, do you need to look at your processing line, is there a problem? And depending on whether your lab's on site or not, it can take, 1 day, 2 days, and time's money. So the people that I work for at the University of Auckland thought that they could make this process a little bit quicker if they used fluorescence spectroscopy. And the idea here was you can stain bacteria to make them fluoresce, and then you can look at the intensity of that fluorescence, and it would indicate something about how many bacteria there are. Now, bacteria are naturally fluorescent if you use UV wavelengths, so if you excite them with UV light, because there's molecules in the bacteria that will fluoresce there, but they are weakly fluorescent, and we don't really like to use UV light because it's a lot more unsafe, right? This is why you wear sunblock. You don't want to be irradiated with UV. So we need a stain, and we need a way to collect our signal. So you all just saw Sam's talk about using his microfluidic device. You'll also see Andrew speak at the end of the session, where he's going to show a quick picture of a centrifugal fluidic device. And these are our groups involved in these projects. And this piece of equipment here is the core to all these projects. Basically, this is our system for exciting and collecting fluorescence light. So we use lasers because they're a nice, intense laser source, light source of a single wavelength, so we can get good fluorescence spectra. We monitor this light source because intensity of fluorescence is proportional to the intensity of the incident light. So to make sure that from day to day we're getting a good signal that's stable and we account for any power fluctuations, we monitor our laser power the entire time. And then we use optical fibers to transport our light around our system so that it's nice and robust, so that you can drop it from a small height, not from a great height, and it's still going to function. It's still going to be aligned. Whereas if we had a free space system, which is a system where you use mirrors and stuff, it's not so robust. So we're always thinking about how can we put this in a factory and have anyone use it. And at the moment, this system has a fiber out here, and then you have how do you interact with your sample. So in the case of Samuel's talk, he would have his microfluidic chip out here with his pump, and you have a fiber or a lens arrangement irradiating the sample, and then another fiber collecting the light back to a detector here so that we can get a spectrum. Why this is a little bit different to a lot of fluorescence examples you'll encounter, because we actually use the spectral data. We're not just seeing how bright is the light. And so for this project, we're staining the bacteria, so we have to look at all the different types of, different types of stain we could use. We're interested in a total viable count, so we want to choose something that's going to stain everything. So we pick something that's going to bind to nucleic acids, obviously, because all bacteria are going to have DNA and RNA. And this is just a sample of 3 that we looked at. They're all excited by using blue light. It's good because it's an easy wavelength to get. It's reasonably safe. You know, if you expose yourself to a blue laser, you blink quick enough that you're not going to do yourself a lot of damage and this sort of thing. And we've gone with this diisodine basically because it has more predictable behavior with concentration of bacteria. Acridine orange is a dye that some people may have heard of in water testing. It's used a lot, but it's got pretty complex behavior with DNA because of the way its structure is, and it can stack and you get all sorts of colors of emission. And what we found, like the general idea was that we were just going to put this dye in, have a look in the easiest sense at the intensity. It would give us an indication of how much bacteria was there. Nothing's ever that easy. What we found was actually, even though this dye is staining just nucleic acids, and bacteria all have nucleic acids, different species of bacteria actually have a bit of a different response to this dye. What you see here is that you probably can guess, gram-positive cells, which have a bit more of a simple cell envelope, are stained a lot more strongly than gram-negative cells. Got to get that around the right way. So we suspect there that, the dye's not getting into the cell as well, so it's not as much dye binding to the DNA, so then you're not getting as strong a fluorescent signal. And this is just a slide showing we've tried a whole bunch of things to try and see, can we get more dye in, what affects the staining. So, if we put EDTA in just at 1 millimolar, did that help? Because that's what a lot of people do with stains. In our case, it didn't. There was no significant difference. If we let it stain for a bit longer, no significant difference. Trying different concentrations of dye didn't really make much difference. So there is quite a strong species bacterial concentration interaction with the dye. And so this is why Samuel's now got his PhD project where we're going to try and look at just single cells or small numbers of cells at once, rather than what's happening in this case is we have a bacterial suspension and we're just putting a dip probe in. So it was like the easiest way you could take a measurement, and it's not quite good enough to give us a very accurate total viable count. The other complexity of it is that word viable, right? So these cyto9 just stains all nucleic acid. Obviously, we don't want to know what's there that's dead. So in this case, you can get other stains. So someone did ask Samuel about this with propydium iodide. It's not cell permeant, so it'll only stain dead cells. When you first get this dye, the company will tell you that cyto9 will only stain live, propydium iodide will only stain dead. It's rubbish. Cyto9 will stain everything. And so what you start to see is then you get this interaction effect, because of course this is 100% live cells, 100% dead cells, and a 50-50 mix. And what you start to see is that when you have a lot of dead cells, you get a lot of cyto9 staining, you get propydium iodide staining, and then what you get is cyto9 will get excited, it will emit, that emitted light will then excite the propydium iodide and inflate its signal. So you get this real interaction that you need to start considering. And so that is another thing that we're having to work on and better understand, so that we can make sure that any count that we do have is just the live cells. So it's definitely still a work in progress, this project. And then just briefly towards the end, just to show you, Raman spectroscopy is my favorite form of spectroscopy, so I like to show it. This was a PGP project done with Steve Holleroyd from Frontera and Michelle Newitt and Cather Simpson from Auckland. And obviously it was, Frontera, there's 2009, the melamine adulteration scandal. So for a few years afterwards, they've still been looking at how do you rapidly see if there's adulterants in the milk. These are just the examples of the small nitrogenous adulterants, or some of them, obviously the ones that up the protein count when you do your Kuldal method. And then you can see very easily here, this is standard milk. You can see these have unique peaks, so this method might have some utility for determining if these adulterants are here. This just shows you that you can spike some milk with these adulterants. You can develop partial least squares regression models, so this is a quantitative method of seeing if adulterant's there. That's not actually that useful if you're developing a method for detecting adulteration, right? You just want to know, is it adulterated in the first case? You don't really care what's there, you just want to make sure, is the milk good to go or is it not? So what we've also done is, this is using chemometric methods. So we're using the whole spectrum, looking at all that chemical information from the milk and from the adulterants and saying, can we see, is this good milk or is it bad? And we're not necessarily saying why it's bad, it's just bad. In this case, these are these nitrogenous compounds, and this is actually sucrose, because it turns out that people do adulterate milk with sucrose quite a lot. And so it shows you that there's, for these spectroscopic methods, like this is just a fiber optic probe, so you could take it out into the field and put it into your milk sample and see very quickly, is it good? Do we take it in? Or do we dump it? Which is of particular interest when you're in, there's a lot of in the Southeast Asian countries or Asian countries where they still use the billy can things, I think you called them, and take them to a drop-off point. It's not quite as, it's not like in New Zealand where the tanker goes out. So then you have those collection points where you need to rapidly test. And so this has some utility there. So this is just to acknowledge all the people involved in the work. This is our team, Fred, Simon, obviously you've all met Samuel, and all the technicians and team that we've had people come and go, and Michelle and Cather. And on this meat project, we used to work with Veritae, but not any longer. Cool. Thank you.